Thank you. Hello, everybody. So I want to start with something that made me go 1997, actually, kind of studying what I'm studying. So what you see here is actually a pretty old movie I'm going to run in a second. And, it's sort of an artist's impression of a very ubiquitous molecular machine that we all have in every cell in our bodies. Every cell on Earth, every living cell on Earth has many copies of these molecular machines. So these objects are called ATP syntheses, and they synthesize ATP. And as all cells on Earth need ATP to function, you need a machine that makes ATP. And these ATP syntheses actually make the energy currency ATP in a very peculiar way. They actually, you can think of them as some kind of generators. So they sit in membranes in the mitochondria, and there's an ion flux that drives rotation, movement in this part of the ATP synthase. And then this causes circular deformations in this part of this molecular machine, and this in turn is being harvested to actually position smaller molecules, the two components of ATP, a designed diphosphate and a phosphate molecule in such the right way that actually you can catalyze a chemical reaction. And it's an uphill chemical reaction that is basically powered from the energy balance by this mechanical motion, which is crazy if you think about it. So this is basically what evolution has invented and optimized over many millions of years, and is sort of, for me, that's an artifact of technology that illustrates what could be done with sophisticated nanotech if you just were able to build something like that. I mean, we've seen some, slightly sort of other versions of nanotech. Sort of this is what biology came up with, and it's just amazing, right? 15 nanometers small, 50,000 atoms, and you get something that just makes, a very sophisticated chemical using a flow. So this is what inspires me. I learned about this as a student, as a physics student, and then started to think about how we can build sophisticated molecular structures using biopolymers. Nature has decided to use amino acid chains and also nucleic acids to build the molecular structures that operate our bodies. And there is a very, very, very powerful principle at play. You can actually encode molecular structures in sequences in biopolymers, and then they self-assemble into the functional three-dimensional states. And the question is, of course, can we harvest this principle for our own purposes? So I spent 15 years in this branch of this research field known as DNA nanotech and DNA origami, and we've made some progress there. Just very quick forward. So we have learned how to build molecular structures encoded in sets of sequences for DNA single strands. So what you see here, actually, this is data. Everything that's yellow and blue consists entirely from design. Basically, it has been built from designed nucleic acid molecules, DNA single strands that self-assemble in aqueous solution into these kind of structures. And all of these, every single object you see here has been validated experimentally using cryo-electron microscopy. So this is not a simulation. This all data was very tediously over many years. Many PhD students work on this to assemble this collection. And there are two actually objects from natural origin here as well. So there's a membrane protein here, 800 kilodalton molecular weight, and there is also a little virus here. This is a hepatitis B virus core capsid. It's 35 nanometers small. And the point of this slide is with sort of modern day DNA nanofabrication, we can build objects that are basically well controlled and of arbitrary shape and size, but commensurate to the dimensions of natural macromolecular complexes. Now, all of these objects are essentially sculptures, and they kind of cannot do anything. They have a certain shape, and I know my aspiration is not to become an artist that makes very sophisticated sculptures. You need a fancy microscope to look at, so to make some of our machines. So last year, actually 2 years ago, we finally succeeded to even build a motor actually using DNA nanofabrication. So there's kind of like a timeline of sort of like some key milestones that we've done in my laboratory. So on the very right, there's a paper cited in 2022. So we made sort of a very small nanoscale electrical motor made from DNA components. Now, the thing that troubled me about DNA nanotech is sort of that DNA itself is chemically inert, basically. It's very hard to actually extract function from these kind of structures. So it's you can easily make nanoscale, very well controlled social frameworks, but you have a hard time to interact with other types of materials, like proteins, for example. These would need to be integrated. So there's the other branch in Bionanotech, which also has been mentioned earlier by Eric, the novo protein design, and this has made rapid strides in the last in the last years. So I'm just reiterating what it actually is. So what is the novel protein design? Well, most proteins in our bodies are of natural origin. The idea is to create entirely new protein sequences from scratch that achieve structural and functional properties that are not found in nature. And one of the key heroes in this field is David Baker. He's been single-handedly pushing this field for many years from very humble beginnings to a great level of sophistication. He runs the Institute for Protein Design, University of Washington in Seattle. And lately we had a number of transformative developments in protein design, which I think were driven mostly by decades of research in structural biology. So as you remember, like AlphaFold, sort of this machine learning tool that was initially developed by Google, was sort of published eventually, and it was trained on all of the hundreds of thousands of crystal structures that have been measured experimentally on proteins, and it sort of knows how a protein looks like, so you can feed it a sequence and it will tell you how it looks like in 3D space. So that's a tool that you can use for structure prediction. And now for protein design, you kind of need to be able to invert that. So define the target structure and extract the sequences that fold into that target shape. And you can actually use AlphaFold for a process called hallucination. Basically, you can start from a random sequence. It generates a three-dimensional structure, which may not even physically fold or be a realistic protein, but you can use that to kind of iterate on the sequence input and then maybe converge in an engineering approach on the target structure. And then there is generative models as well that have been mentioned previously. So the dream of creating protein function from scratch has become so realistic now. There's also has been made sort of significant investments in the commercial space. So Therapeutics, for example, is a company that was launched with $1 billion seed money, which is an enormous amount of money. So Altos Labs, which was mentioned earlier, also got $3 billion in seed funding, but sort of just same scale. And then it's also recently a new company, Evolutionary Scale, got seeded with $142 million investment. The idea here is that you can create therapeutic proteins from scratch. Now, we also have been actually stealthily active in this space. So we came up with our own protein design pipeline, which will soon appear in a major journal. And it is something that actually implements a process which is useful for engineering, like a gradient descent-based optimizations, very fast. You can build basically very large proteins. I don't want to go in data, but just showing this one. So the green is a design structure, and the overlay in gray is actually the experimentally measured structures. And this is a 1,000 amino acid large protein. This is as large as an antibody. It's the biggest de novo designed protein that's ever made. And so I'm happy that we have this sort of unique pipeline, which we can add to the portfolio of protein design tools. There's another one we made. Actually, I think I'm kind of running out of time, so I want to close this talk by basically, we're going full circle, kind of. We have now design tools available, which allows us to design proteins in arbitrary shapes. So we just, for fun, we spelled DNA in proteins. So for me, that's sort of full circle, trying to complete all the materials. So where do I think this will go? Well, eventually we have these three areas of Bionanotech, DNA nanotech, the novo protein design, RNA nanotech. They each have their specific advantages and also kind of some limitations. So eventually we'll have hybrid technology, just combine basically the advantage from each of these fields to make something that works. I kind of personally disagree slightly with Eric that design needs to be accelerated. I think experiment needs to be accelerated. So I spent 15 years of my life. I'm going to age quickly if this progress is that slow. And I think we're bottlenecked by experiment. It's too hard to validate structures, too hard to test the function with the sort of the manufacturing kind of methods that we have in the lab. So I'm thinking very hard about how to solve that problem. And we have another problem, which is kind of, we have this dichotomy of academic creativity, where you can come up with almost anything and make a very complicated, like a Rube Goldberg machine that does something. And then there are the market forces which tend to push things into a very simple, much more simple, sort of economical viable state. And these things often don't really go well together. So you can make advanced therapeutics that has basically no future in the real world because it's just too expensive, but it would still be useful for maybe humans. Okay, that was it. Thank you very much.
Thank you. For a quick question? Yes, go for it.
What are the major bottlenecks for in silico verification for scalability?
In silico verification, well, I guess there oftentimes basically you don't have enough data to build on. Like this is no longer the case, I think, for a certain fraction of sort of the design targets that you would do in protein design, because so much structural data went into there. But you don't have, for example, a comparable level of data in DNA nanotech or RNA nanotech, so there's very little data. You cannot train an AI on the data. And then what I'm personally not so happy about with the AI methods is it's hard to extract kind of the underlying physics of it. You have this model, you run something through, get something out, and you don't really know why. It's kind of whereas if you have actually a physical model where you know what are the forces at play, you can write down an equation, you have a better chance of sort of understanding what's going on. So the design, I think design is bottlenecked by experiment is bottlenecked by design, so this is like an iterative thing. And so you need to be better in all of these camps. Eric, you have a question?
If design produced really compelling targets for experimental work and got you, I don't know, 1 or 2 orders of magnitude more funding for that experimental work, would the world look a little different to you?
I guess it would be better. It would not be perfect yet, because you still would have to do the experiment, because sort of, in the end, the test of all knowledge is experiment. You have to be able to do the experiment and show that this thing is working. And if it's too small, for example, then it's too hard to make it, right?
Being able to report parallel experiments, unlimited access to bioreactor?
Yeah, that would be pretty good.
I would think it'd be pretty good.
Yeah, that would be pretty good. Yeah. And then the function, yeah, exactly. The function, like for example, driving a chemical reaction, you have to do all the, the analytics and things like that. It just takes a lot of time and people.